ASH is known for its great science, and this year's annual meeting is no exception. I'm joined now by Dr. Ebert and Dr. Lentz, and we're going to talk about the best of ASH. We're going to preview this session. So what were the best abstracts?
We were really thrilled by the quality of the abstracts this year. There were so many outstanding abstracts, and we took a great amount of time and deliberated about what some of the best abstracts were, but we chose some themes that were really highlighted some of the best abstracts at ASH, one of which was immunotherapy. So a variety of different approaches to using the immune system to target cancer have really come to fruition now. One being anti-PD-1, this is called a checkpoint blockade inhibitor that allows the immune system to attack leukemia cells or lymphoma cells. Another is engineering T cells to target cancer cells, that's called CAR T cells. And a third one is called BiTE cells, or bispecific antibodies that target leukemia cells as well. And these have been developed over many years, but really are coming to fruition now and having the clinical benefit. And it's been exciting to see a suite of abstracts that do those, that use these mechanisms. Another theme is genetics. There are many abstracts here about the genes that are mutated in hematologic malignancies, but there are also some unusual use of genetics, such as using sequencing to find circulating DNA that is from a lymphoma, but circulating in the bloodstream that can be used to detect minimal residual disease. Another is combining germline and somatic mutations, not just looking at the genes that are somatically mutated in cancer, but how they integrate with germline mutations that predispose patients to cancer. And another is looking at clonal mutations that happen before cancer arises, a preclinical state for malignancy. Another theme that I'll add is epigenetics. Many of the mutations that lead to malignancy and the various hematologic malignancies alter the epigenetic state of cells, alter their methylation or histone marks. And I'm going to highlight in the best of ASH several abstracts that alter DNA methylation, a new base called 5-hydroxymethylation, and potentially what the therapeutic implications of those are.
And Dr. Lentz, would you like to expound on some of those?
A couple other things I'll mention is that some of the best abstracts that I'll be talking about have to do with genetic or genomic therapies for some hematologic disorders such as hemophilia, which is starting to take off now and enter into the clinic. And also there's great progress being made on gene therapy for thalassemia and sickle cell disease with some really novel approaches using genomic engineering techniques like zinc finger proteases. I also plan to highlight a few abstracts in relation to anticoagulation therapy and platelet biology. One of the plenary presentations yesterday was on platelet clearance by the Ashwell-Morel receptor and how that regulates thrombopoietin production and feeds back into mechanisms of regulation of platelet production, which has implications for several disorders within the hematology spectrum, including myeloproliferative disorders, transfusion medicine, and even ITP, where we're learning that one of the mechanisms of antiplatelet antibodies in ITP has to do with desilylation of platelets and clearance by the AMR. And then finally, I'm going to highlight one of the areas that I think is the most interesting new developments over the last few years in hematology, which is what we've learned about the regulation of iron utilization and iron metabolism. We heard in the Donnell Thomas lecture today from Tom Gantz great progress in understanding hepcidin and erythropoietin, ferrone and ferroportin, and we're really on the cusp now of having therapeutics developed from these new discoveries, so that I think very soon hematologists will have a new toolbox to address disorders such as iron overload and anemias related to iron. So those are some of the things that I'll be highlighting.
Yeah. And what really makes these abstracts the best of the best at ASH 2014?
I guess, as Steve said, one of the things that we tried to do was to collect themes and fields that have really come to fruition in the past year. And we tried to combine things that are scientific breakthroughs that also have clinical implications. And so maybe fields that have been evolving over many years but finally have come to fruition. We really understand the science very well, and these findings will influence the care of patients.
Yeah, I think one of the great things about the ASH meeting is it's a great scientific meeting, but it also makes the science relevant to the practicing hematologist. And I hope the Best of ASH session will be valuable to both scientists and clinicians.
Thank you both so much for your time. I'm Rachel Kopchak for ASH News TV.